Bueno, buenos días a todos. Vamos a proceder al acto de defensa de la tesis doctoral de Francisco Javier Álvarez Martínez, que opta al título de doctor en Biología Molecular y Celular por la Universidad Miguel Hernández. El título de la tesis doctoral es Compuestos antimicrobianos de origen natural, una oportunidad para el tratamiento de enfermedades infecciosas resistentes a antibióticos. Antes de proceder al acto de defensa, el secretario del tribunal, el doctor Luis Pérez García Están, va a mencionar algunas cuestiones legales. Posteriormente yo voy a presentar al tribunal y después te daremos la palabra. ¿De acuerdo, Javier?
De acuerdo.
Luis, cuando quieras.
Muy bien, gracias. Bueno, pues como decía la presidenta, esta tesis también opta a la mención de doctorado internacional por la UMH. La presentación de la misma será en lengua inglesa. Esto es una sesión pública y va a ser grabada lo que es la presentación, pero no así el turno de preguntas posterior, que en principio hemos quedado que lo haremos en castellano. Según la normativa, la presentación no debe exceder de los 60 minutos y el doctorando deberá mantener encendida su cámara web y su micrófono durante la totalidad del acto. No puede dejar de estar conectado. Se ruega al público asistente que tenga los micrófonos desconectados. Para el tribunal es opcional, pero bueno, si tenemos miedo de que nos pueda sonar un teléfono o algo, pues también, y si no, para nosotros es opcional. Y nada más, cuando te cedo la palabra también, Juncal.
Bien, bueno, pues nos presentamos un momentito. Estamos 3 personas en este tribunal de tesis. El secretario, como acabo de mencionar previamente, es el doctor Luis Pérez García Están, que es profesor titular de la Universidad Miguel Hernández de Elche, en el Departamento de Bioquímica y Biología Molecular. Como vocal tenemos al doctor Pedro Miguel Mena Parreño, que es Assistant Professor en el Departamento de Ciencia de los Alimentos y del Fármaco en la Universidad de Parma, y como presidenta yo misma, que soy Juncal Garmendia García, científico titular del CSIC en el Instituto de Agrobiotecnología en Navarra. Vamos a proceder a la presentación. Just briefly, I will just quickly switch to English because we know that we have a person who is not sleeping today. We just presented ourselves, and Javier is going to present his work in English, and afterwards we will have the discussion and the dissertation in Spanish. So thank you very much for attending to the presentation, and now we are going to switch into English for the presentation. Okay? Javier, cuando quieras.
Good morning. My name is Francisco Javier Álvarez Martínez, and before I begin, I would like to deeply thank the members of the tribunal who have agreed to be part of this defense today. Thanks also to my 2 thesis directors, and thanks to all the people who are today supporting me here. And now, with the permission of the tribunal, I'm going to present the defense of my doctoral thesis entitled Antimicrobial Compounds of Natural Origin, an opportunity for the treatment of infectious diseases resistant to antibiotics, that was directed by Dr. Vicente Micol Molina and Dr. Enrique Barrajón, Catalán. This presentation is structured in 6 different parts, being first, introduction, second, objectives, third, methodology, 4th, results, 5th, conclusions, and 6th, annex. Let's start with the introduction. I would like to begin with this quote from Selman A. Waxman, a leading scientist in the field of antibiotics, who won the Nobel Prize in 1952 in medicine for the discovery of streptomycin. And he said, "In learning to utilize antibiotics for the control of human and animal diseases, the medical and veterinary professions have acquired powerful tools for combating infections and epidemics." And this is a great truth, since it shows that the antibiotics are just a tool that must be used appropriately. Not, they will not be as effective and will end up losing their usefulness. Here, in this diagram, we can see when the different families of antibiotics were discovered. Now, the arrows below indicate when bacteria resistant to the antibiotic family of the same color were identified. To date, resistant bacteria have been found against all families of antibiotics. And to further aggravate the problem, we can see that no new classes of antibiotics have been discovered recently. So we don't have new tools to fight against the antibiotic-resistant bacteria. And what are the main causes of the antibiotic resistance rising we are living? Well, first, among the main causes, we have the antibiotic overprescription and also the self-medication. Second, we have the early abandonment of treatments, which can lead to more resistant recurrent infections. Third, we have the excessive use in aquaculture and livestock. This is very important because hundreds of tons of antibiotics are used every year and end up entering the biosphere, producing a great selective pressure on bacteria in the environment, favoring the emergence of resistances. Fourth, the uncontrolled infections in health centers. As many sick people are concentrated in these centers, caution must be extreme. Fifth, lack of hygiene. And this problem is especially serious in certain developing countries. Sixth and last, it's the shortage of discovery of new antibiotics, as I said before. And one of the main causes for that is that companies see the investment in antibiotics as a risky bet, since if resistant bacteria emerge shortly after the production of a new antibiotic, it will be very difficult to recover the large economic investment required to produce these new antibiotics. And to see the problem of the antibiotic resistance, let's imagine that this is a pathogenic bacteria that infects a body. It starts to divide, and to divide. Eventually, through a spontaneous mutation or through the acquisition of exogenous resistant genes, it can acquire resistance to antibiotics. If we decide to treat this infection with an antibiotic, we will eliminate the parts of the population that is sensitive to the antibiotic. However, that resistant population can endure. And in this way, this resistant bacteria can again divide, and divide. And the antibiotic will cease to be effective against the infection, causing a major health problem. Among the most important mechanisms of resistance to antibiotics, we have the expression of efflux pumps that are responsible for expelling the antibiotics out of the bacteria, the inactivating enzymes, which are responsible for transforming the antibiotics into inactive molecules, the decreased uptake, which consists of a permeability decrease of the cell wall and the membrane, so less antibiotic goes inside the bacteria. And also we have the target alterations, which consists of transforming their molecular targets so the antibiotic cannot recognize them and carry out their action. And regarding the methods of acquisition of antibiotic-resistant genes, this can occur by transduction via virus, conjugation via plasmid, spontaneous mutations, or finally by transformation with exogenous free genetic material. Now I would like to show this video that is from Harvard Medical School, which I think it's a brilliant way to illustrate how the adaptation to antibiotics work. In this test, a giant rectangular petri dish was used, in which E. coli, sensitive to ciprofloxacin, was cultured on both ends of the petri dish, and increasing concentrations of ciprofloxacin were banded towards the center of the plate. In this video, which is very accelerated, it is observed how clearly the bacteria managed to adapt and colonize the bands of the plate with high concentrations of the antibiotic. Bacteria took 11 days to grow through the entire plate, colonizing the central area that had 1,000 times the initial concentration of ciprofloxacin. So they have acquired a very high level of resistance in just 11 days. So I think it was very illustrative, very good example and design. And to give specific data on the magnitude of the antibiotic resistant problem, we have the example of the USA, where we have 2.8 million antibiotic resistant infections per year, resulting in 35,000 deaths per year. Among the most troublesome and widespread antibiotic resistant bacteria in the world is methicillin-resistant Staphylococcus aureus, known as Mrsa. It is considered a superbug, as it is resistant to most of the antibiotics commonly used to treat the infection it causes. It is also related with higher morbidity, mortality, and length of stay in patients affected by this bacterium. It results in a very high healthcare cost impact. And as I said, it's very common, and it's very widespread throughout the world, and it's very prevalent both in hospital and community environments. Here, we can see the prevalence of MRSA in Europe. There exists a very clear prevalence gradient that can be seen from north to south, where the incidence of these strains is higher. The data from Spain is worrying because although it appears in the orange prevalence here between 10 and 25%, its specific data place it very close to the 25% and with an upward trend. So, given this scenario, what's the strategy we propose to deal with these antibiotic-resistant bacteria? Well, the strategy we propose is to use plants as a source of bioactive molecules with antibacterial activity against the antibiotic-resistant bacteria. Plants have been used as a therapeutic tool for thousands of years. Proof of this is the Eber papyrus that dates back to 1500 before Christ and reveals that at that time, the medicinal properties of plants such as Ricinus communis were already known. Throughout history, plants have been used in very different forms, in different ways. Poultices, extracts, infusions, oils, etc. One of the advantages of using plants as a therapeutic tool is that they are very abundant in our planet, reaching 80% of the entire terrestrial biomass. In addition, its metabolism can produce a huge variety of different biomolecules, many of which have been shown to have beneficial effects on human health. One of the groups of biomolecules of plant origin that have attracted the most attention in recent years are polyphenols. To date, more than 80,000 different polyphenols have been identified, and these can be subdivided into subclasses, depending on their chemical structure. These subclasses are phenolic acids, lignans, flavonoids, stilbents, and hydrolysable tannins. As I said, polyphenols are plant molecules that have one or more phenolic groups. They are part of the secondary metabolism of plants. Their function is defense against stressful situations in plants. They are very abundant in fruits and vegetables present in the common diet of humans. They have beneficial effects for the human health. And they have a very special characteristic known as molecular promiscuity. And we are going to expose this in depth with a graphic sample. Let's take the example of an antibiotic like penicillin G, whose activity is not molecularly promiscuous. This antibiotic acts on a specific target, in this case, the penicillin binding proteins located inside the bacterial cell wall. If this target changes, like in here, the antibiotic is no longer effective. In contrast, polyphenols do not act on a single target. They can act on multiple different molecular targets, hindering the potential development of specific resistance mechanisms. Among the most active polyphenols identified to date, we have camphorol, myricetin, quercetin, and punicalagin, being the camphorol, myricetin, and quercetin flavonoids, and the last one, punicalagin, being an hydrolysable tannin. Let's move to the objectives. The first main objective of this doctoral thesis was to characterize the antimicrobial capacity of different compounds of natural origin. Second was to deepen the knowledge of their mechanisms of antibacterial action and their molecular targets. And third was to evaluate the suitability as an alternative or a support to existing antibacterial therapies. Regarding a methodology, here we can see the methodology followed for the extract production. First, we have the collection process, in which Cistus albifolius, using the extract, was planted here at the Miguel Hernández University, while the Punica granatum extract was purchased from Monteloeder Company. Regarding the extraction, we did the extraction of the cystus in an aqueous media at 55 degrees Celsius for 4 hours with an 1:5 ratio, 1 gram of plant, 5 milliliters of solvent. These extraction conditions were optimized during my final master's work, in which dozens of different extractions were carried out using different organic solvents, different temperatures, times, ratios, and also using technologies such as microwaves, extraction assisted by ultrasounds. And part of this work was carried out during a short stay at the Polytechnic University of Valencia. Finally, it was determined that the optimal conditions to obtain the richest possible extracts in polyphenols were those used in this doctoral thesis. Then we have the fractionation that was performed by column chromatography in a purification process using Amberlite FPX66 resin. And lastly, the drying process that was performed by a spray dryer machine, model Bucci mini spray dryer B290. These are the material methods used for the molecular characterization and the biochemical tests that we use to characterize the activity of the extracts. First, we have the following that was used to determine the total phenolic content using the gallic acid equivalent method. Then we have the TIAC, that stands for Trollocs equivalent antioxidant capacity, that was used to determine the antioxidant capacity by comparing to a standard that was Trollocs. These two tests were performed in 96 well plates. Then we performed the high-performance liquid chromatography coupled to mass spectrometry to characterize the molecular composition of the extracts. We used an Agilent 1100 series HPLC coupled to an Squire 3000 plus mass spectrometer. Regarding the antimicrobial tests, we performed two different tests. The first being disk diffusion assays tests in Petri dishes, and also we performed microdilution assays in 96 well plates. And for the data analysis, we used different softwares, being GraphPad Prism 6, Microsoft Excel, and Google Colab, some examples of them. Moving to the results section. Chapter 1 of this doctoral thesis corresponds to the review article entitled Antimicrobial Capacity of Plant Polyphenols Against Gram-Positive Bacteria, a Comprehensive Review, published in Current Medicinal Chemistry. Its authors are Enrique Barrajón, Catalán, José Antonio Encinar, Juan Carlos Rodríguez Díaz, Vicente Micol, and myself. My role in the article consisted of searching the bibliography, writing the work, and coordinating it. José Antonio Encinar was in charge of carrying out the molecular docking part, in which I assisted as a collaborator. This review focuses on the activity of polyphenols on gram-positive bacteria and shows that there are exceptionally active compounds against them, including against resistant strains. Polyphenols with more prominent activity include unicalagen, myristicin, magnolol, quercetin, camphorol, and honokiol. They have shown a minimum inhibitory concentration, MIC, of between 4 and 16 micrograms per milliliter against S. aureus. One of the most interesting findings was that polyphenols show synergistic activity with other polyphenols and or antibiotics of clinical use. One of the most effective polyphenol combinations is punicalagin together with lactic acid against S. aureus. And this synergistic activity can even sensitize antibiotic-resistant bacteria and make them sensitive to the antibiotics. For example, the combination of magnolol or honokiol together with antibiotics showed a great efficacy against clinical isolates of Mrsa. So we have that polyphenols plus another polyphenol or antibiotic can have a greater combined antimicrobial effect. Another of the key findings of this review lies in the molecular promiscuity of polyphenols that allows them to act against various different molecular targets. We can see here in this figure. Among the mechanisms and targets, we have the cell wall, cell membrane, cell wall proteins, membrane proteins, the ATPase, the DNA, the RNA-related molecules, cytoplasmic proteins, and nutrients and ions outside the cell. And among the multiple mechanisms of action and targets identified to date, it appears that the most common polyphenolic targets are the cell wall and the bacterial membrane, the two first. In addition to the review part of the article, this work has an original section on computational molecular docking, carried out in order to identify polyphenols with great antimicrobial potential. For this test, the 3D structures of nine enzymes involved in the biosynthesis of peptidoglycan of the bacterial cell wall were used. Here we have the 8 firsts with the structures and inhibitors inside their structure. And this last enzyme, the NIND, is the glutamate racemase, also known as Mrsa, and has been widely studied as a pharmacological target in recent years. We will use the data of the docking against this enzyme here as an example in the next slide. And 931 polyphenols present in the Phenol Explorer 3.6 database were used as ligands for the molecular docking against the target enzymes. The test showed that polyphenols have great inhibitory potential for glutamate racemase activity, and it would be interesting to develop in vitro tests to corroborate this activity. I will present this graph as an example of the molecular docking against this enzyme. Here, I will make it bigger to see it better. The different Gibbs free energy is here, and these values are calculated for a section of polyphenols against the Mrsa target of different bacterial species that we have in here in different colors. On the left, in yellow color, we have the inhibitors in the bibliography. So they are reference inhibitors, and this part is are the polyphenols with codes. And the lower the Gibbs free energy, the higher the binding affinity of the ligand with the enzyme. We can see that there are polyphenols that show a high affinity for this target, even greater than the reference inhibitors found in the literature, which can be seen on the left of the graph, as I said, in yellow color. Among the most active polyphenols are here these 2 thiaflavins in green color, here, and also 6 catechins, here 1 and here 5 rest of them, marked with the orange color. So these compounds were very promising and deserve further assays to confirm this inhibitory potential. In addition to the in silico test included in this review, during the doctoral thesis, other molecular docking works were carried out. One of them is the following, which was presented at the Congress of the Infectious Diseases Society of the Valencian Community in 2017. This work, a screening of thousands of natural molecules, was carried out against the PBP2A protein, found here, of Mrsa, which is the one that confers the resistance against the antibiotics such as methicillin, penicillin, and their derivatives. In this work, more than 67,000 different natural compounds extracted from 13 different databases of chemical structures were screened as possible inhibitors of PBP2A. In addition to the free energy calculation, we also performed a pharmacological test to measure the suitability of these compounds to be developed as drugs, measuring the Lipinski's rule of 5 and their ADMET parameters. Here are the pharmacological properties filter that we used. Moving to the results and conclusion section of this poster. Finally, 2,820 molecules passed the first screening. These were subjected to various filters and structural groupings to eliminate the redundant compounds and select those who were the most promising. Finally, just 3 compounds were selected as the most interesting, as they went on to the in vitro testing phase. These 3 compounds were purchased and a pilot testing process was started to fine-tune the in vitro test at the Alicante General Hospital using Mrsa isolates with the PBP2A protein. The first approximation did not obtain clear results, probably due to the lack of optimization of the test and the repetition of these essays pending as future work for this line of research. Chapter 2 of this doctoral thesis corresponds to the article published in Scientific Reports entitled "The antimicrobial capacity of Cistus albifolius and Punica granatum extracts, plant extracts, against clinical pathogens is related to their polyphenolic composition," whose authors are Juan Carlos Rodríguez Díaz, Fernando Borrás Rocher, who oversaw the statistical section together with Enrique Barrajón Catalán, Vicente Micol, and myself. My role in this article consisted of conducting the antimicrobial tests, analyzing the data, and writing most of the article. In this work, a first screening was carried out using 3 different plant extracts and 7 pure polyphenolic compounds against 11 different bacterial species of clinical relevance. The antimicrobial tests were carried out using the disk diffusion method. The table shows the percentage of isolates that were sensitive to each compound used. We can see that the most active compounds were the extract of Cistus albifolius, CS, Cistus from now on, and Punica granatum, pomegranate, from now on, and that the bacteria most sensitive to their action was S. aureus. Both extracts and the Staphylococcus were selected for conducting the second part of the antimicrobial tests, now in microdilution method with 96 well plates. Punicalagin, here P, was also very effective, but as it is the main component in both of the extracts, we decided to go ahead and to move selecting the 2 extracts as they had its activity covered. The molecular composition of the Cistus and pomegranate extracts was determined by high-performance liquid chromatography coupled to mass spectrometry. Here we can see the chromatograms. Here, the first for the Cistus extract, we can observe that the main compounds, white peaks, are the punicalagin, myrisectin exoside, and quercetin glucoside, meaning that the extract mainly contained hydrolysable tannins plus flavonoids. Regarding the pomegranate extract, we can see that the 2 most abundant compounds were punicalagin isomers, meaning that the whole extract was mainly composed of hydrolysable tannins. For this second phase of antimicrobial tests, 100 clinical isolates of S. aureus were selected and collected from patients by the Clinical Microbiology Service at the Alicante General Hospital. Of these isolates, 50 were methicillin-resistant strains, and the other 50 were methicillin-sensitive strains. In the present box and whisker diagram, we can observe the results of the MIC50 values of the extracts of Cistus in green color, here, and the pomegranate in orange color, against the resistant in the left part, and the sensitive isolates on the right. Here, the excess inside the boxes correspond to the mean MIC50 value, which in all the cases were between 50 and 80 micrograms per milliliter. We also can observe that the MIC50 values of the cistus extract are significantly lower against the resistant strains than against the sensitive strains, which shows a greater activity against the resistant ones. In addition, we can see that the pomegranate extract was more effective against the sensitive strains than the cistus extract. Here, we have a very big table with a lot of data. And it is that the Clinical Microbiology Service at the Alicante General Hospital carried out a study of the antibiotic resistance profile for each of the 100 S. aureus isolates against 16 different antibiotics commonly used in clinics. The antibiotic resistance here in this table is represented in red squares. The intermediate resistance is in yellow, that there are very few ones, like here and here and here. And the sensitivity is in green. In this slide, we can see the resistance profile of the 50 methicillin resistant isolates, numbered from 1 to 50. In this slide, there is the profile of the isolates sensitive to methicillin, numbered from 51 to 100. We requested this data of the previous table to perform the following statistical analysis. The MIC50 values for each extract in the upper part for cistus and the bottom for pomegranate was then grouped based on the resistance. Here are the squares, the resistant, or sensitivity, the circles, here, circles, of the isolates to each antibiotic. In this graph, we can see that the cistus extract was more effective against resistant bacteria, as the squares are in general below the circles. In the pomegranate extract below, it seems that it was more effective against the sensitive isolates, as the circles are in general below the squares. Analyzing the significant differences, the cistus extract showed significant differences for the beta-lactam antibiotics, methicillin and oxacillin, being more effective against the resistant isolates. So we can see here, the resistant, the squares, are below the circles, so they have the less MIC50 when we do the mean values. Moving to the pomegranate extract, it showed significant differences against isolates sensitive to oxacillin. Sorry. It's not working, the laser pointer. Okay. Don't worry. Showed statistically differences against the sensitive to oxacillin and the quinolones that include the ciprofloxacin and levofloxacin. It was the contrary case from the cistus extract. To corroborate the results observed in the previous tests, a multiple correspondence analysis was carried out, which serves to determine which variables contribute the most to the differences between the samples and capture the results in a two-dimensional graph, like the one observed here in this slide. The different groups can be observed based on the grouping of the data for each individual isolate. Resistants are in red, and sensitives are in blue. The A group is an heterogeneous group of isolates with resistances of up to 4 antibiotics, but without common characteristics, as they are very close to the origin point. B are isolates sensitive to most of the tested antibiotics, and it can be considered the most sensitive population in this study. C contain just 9 isolates, also presenting sensitivity to most of the antibiotics as in group B, but 8 of them show resistance to erythromycin and clindamycin. The D group contain only 6 isolates, and up to 5 share the resistance to beta-lactam antibiotics. Group E includes 15 isolates that have common resistance against beta-lactam and quinolones, plus erythromycin, resistance in 14 of them. Finally, the F group, that was the most interesting group for us. It contained 25 isolates with resistance against beta-lactam antibiotics and quinolones. And 17 of the isolates in this group were among the 50 most sensitive isolates to the cistus extract. This observation reinforces the idea that there is a link between the antibiotic resistance profile and the antimicrobial activity of these extracts. This other multiple correspondence analysis, it was done using the categorical values instead of the individual data as before. The values were grouped according to the resistance in red color, the intermediate resistance in black color, we have some examples in here and here, or sensitivity in blue color to each of the antibiotics. The trend that we can observe in this graph is that the isolates sensitive to the antibiotics are very close to the horizontal axis, while the resistant ones, the red ones, are grouped in the right part of the graph. This trend is reversed for the clustering of the MIC50 values for the cistus extract. The group of isolates most sensitive to the cistus extract is found near the area of the isolates most resistant to antibiotics, reinforcing the idea that the cistus extract is more effective against isolates resistant to antibiotics. The chapter 3 of this doctoral thesis corresponds to the review article entitled Tackling Antibiotic Resistance with Compounds of Natural Origin, a Comprehensive Review, published in Biomedicines. Its authors are Enrique Barrajón, Catalán, Vicente, Micol, and myself. My role in this article consisted of searching the bibliography, writing the work, and coordinating it. In this work, we broadened the search spectrum for natural antimicrobials. We went from focusing only on compounds of plant origin to exploring sources such as animal, fungi, and bacteria, sources of biomolecules with antibacterial capacity. And we had special interest in their activity against the bacteria resistant to antibiotics, the drug-resistant bacteria. We found that the majority of naturally occurring compounds with antimicrobial capacity, regardless of their origin, do not usually have enough therapeutic power to develop monotherapies based on them against antibiotic resistant to bacteria resistant to antibiotics. Nevertheless, the application of this in combination with antibiotics of traditional use has been very effective in recent studies, making in many cases that the traditional antibiotics recover or even increase their lowest levels of effectiveness against the resistant bacteria. These findings are very similar to those described in Chapter 1 in reference to compounds of plant origin. So we can conclude that the combination of antimicrobial compounds of natural origin and antibiotics has a therapeutic potential that can be explored in depth. In this figure, we can see the different mechanisms of antibacterial action of various compounds of natural origin. At the top right, there's a color legend in which we can see that the orange compounds are of animal origins, blue ones are bacterial origin, the gray ones are fungi origin, and the green ones are of plant origin. We can observe that most common mechanisms of action of these compounds are related to the disruption of the protein biosynthesis, the cell wall, and the plasmatic membrane. Let's make it a bit bigger to see it a bit better. Regarding the compounds that act on protein biosynthesis, the compounds of bacterial origin clearly predominate in this group, as the majority are in blue color. The compounds of bacterial origin also predominate in the groups of compounds which disrupt the cell wall, although there is a bit more of diversity than in the previous group. Finally, the compounds of plant and animal origin abound in the group with the mechanism of disruption of the cytoplasmic membrane. So there is a bit more of diversity. Regarding the bacteria developing resistance to natural compounds, there are some recent studies that suggest that some bacterial isolates can develop certain levels of resistance against plant compounds. Nevertheless, the mechanisms of resistance behind these observations remain unknown, and the literature of the subject is very scarce. Clarifying this question was the main objective of my research stay that I carried out during the doctoral thesis at Carleton University in Canada, under the supervision of Dr. Alex Wong. Unfortunately, that line of work had to be paused due to the current pandemic, and I hope it can be resumed soon. As a final finding in this chapter, we observe that there is a growing trend both in the discovery of new antimicrobial compounds of natural origin and in the discovery of new properties in already identified compounds. And this trend is mainly attributed to the advancement in the use of new technologies such as the omics, the network pharmacology, the informatics, etc. In this figure, it can be seen how the publications in PubMed related to antimicrobial capacity, antimicrobial compounds, grow year after year, with their trend being upward. And now, with the permission of the tribunal, I will proceed to read the conclusions of the present doctoral thesis. First, there is a lot of evidence of natural compounds that have proven antibacterial capacity, some being effective against bacteria resistant to antibiotics, especially those responsible for nosocomial infections. Among natural compounds, plant polyphenols represent an antimicrobial therapeutic arsenal that hasn't been fully explored yet, either in isolated form or in combination with antibiotics. Third, in general, polyphenols have greater antimicrobial activity against gram-positive bacteria than gram-negative bacteria. The mechanisms of action of polyphenols are varied and multifactorial. disturbance of the cell wall and or the plasma membrane, alteration or modulation of membrane receptors or ion channels, inhibition of bacterial metabolites, or biofilm formation. It is postulated that the perturbation of the structure or of the synthesis of the cell wall and or the lipid membrane is the predominant mechanism for most plant polyphenols. Fifth, given their multifactorial nature and molecular promiscuity, the study of the natural compounds and antimicrobials must be approached through polypharmacology, network pharmacology, and computational virtual screening. This will increase the effectiveness of new compounds, reposition already known compounds, and reduce possible side effects. 6. Plant extracts rich in polyphenols from Cistus albifolius, rich in hydrosoluble tannins and flavonols, and Punica granatum, rich in hydrosoluble tannins, have been shown to possess antibacterial activity against clinical isolates of Staphylococcus aureus, including Mrsa strains. 7th, the susceptibility of the clinical isolates of the Staphylococcus aureus to the plant extracts of Cistus albifolius and Punica granatum is related to the antibiotic resistance profile of each isolate. The Cistus albifolius extract is more effective against clinical isolates of S. aureus resistant to beta-lactam antibiotics, while the Punica granatum extract is more effective against quinolones and oxacillin-sensitive isolates. 8th, there is a synergistic activity between different classes of polyphenols and between certain polyphenols and clinical use antibiotics. Combinations between flavonoids and beta-lactam antibiotics have shown particularly high levels of synergy. And 9th and final conclusion of this doctoral thesis is that the use of combined antibacterial therapies using plant extracts or polyphenols together with antibiotics show potential advantages, such as minimizing the risk of resistant development and a lesser occurrence of side effects for patients. According to the results obtained in the present thesis, the knowledge of the composition of the studied extracts or mixtures and their antimicrobial action would allow the development of individualized therapies according to the antibiotic resistance profile of the bacterial strains to be treated. And before I finish, I would like to deeply thank to the people and the places where I have been doing my doctoral thesis. First, the IDIBE here at the Miguel Hernández University, under the supervision of Dr. Vicente Micol and Dr. Enrique Barrajón. The Alicante General Hospital under the supervision of Juan Carlos Rodríguez Díaz. The NIO, the Netherlands Institute of Ecology, where I did another internship under the supervision of Dr. Olaf Tick and Dr. Paulina Garbeba. And also the Carleton University, the biology faculty, under the supervision of Dr. Alex Wong. Thank you very much to you all. And also, gracias, thank you to my research group here at the IDIB. So thank you for your attention.